Light reactions take place during the first stage of photosynthesis. During this stage, light energy is converted into chemical energy in the form of ATP and NADPH, and hence it is also called the photochemical phase. It takes place in the thylakoid membranes of the chloroplast and includes photosynthetic pigments arranged into two light harvesting complexes within photosystem 1 and photosystem 2. Light harvesting systems, or LHCs, also called antennas, are made up of many pigment molecules except a single molecule of chlorophyll A. These pigment molecules are bound to proteins and absorb different wavelengths of light and help increase the efficiency of photosynthesis. The single chlorophyll A molecule forms the reaction center in PS1 as P700 and in PS2 as P680, symbolizing the absorption maxima of chlorophyll in these centers. Light reaction consists of the following four stages: light absorption, water splitting, release of oxygen, and formation of high-energy intermediates ATP and NADPH. In the light absorption stage, the chlorophyll A in PS2 absorbs the 680 nm wavelength of red light. This causes excitation of electrons, thereby attaining high energy levels. These electrons are picked up by an electron acceptor, which transfers them to the electron transport system. The electron transport system consists of cytochromes. The movement of electrons is downhill in terms of lower redox potential. The electrons from PS2 are passed on to PS1 through the electron transport system. Simultaneously, electrons of the PS1 reaction center are excited by absorbing red light of wavelength 700 nm. These are transferred to another acceptor molecule with greater redox potential. These electrons move downhill to a molecule of energy-rich NADP+, thereby reducing it to NADPH, forming a Z scheme. In water splitting, water is split into H+, oxygen atom, and electrons. The electrons that were removed from PS2 are replaced by electrons formed due to the splitting of water. The electrons needed to replace those removed from PS1 are provided by the excited electrons of PS2 through the electron transport system. Water splitting complex is associated with PS2. The process of synthesizing high-energy compounds like ATP by cells in the chloroplast and mitochondria is called phosphorylation. In non-cyclic photo, phosphorylation PS2 and PS1 work in series. These two photosystems are connected through the electron transport chain. ATP and NADPH+ are synthesized in this electron flow. The cyclic flow of electrons occurs in the stroma lamellae membrane, where only PS1 is functional. This is because the lamellae membrane lacks both PS2 and the NADP reductase enzyme. The excited electrons do not pass on to NADP+, but are cycled back to the PS1 complex through the electron transport system. This cyclic flow results in the synthesis of ATP only. Cyclic photophosphorylation also occurs when only light of wavelengths beyond 680 nm are available for excitation. The chemiosmotic hypothesis explains the mechanism of ATP synthesis. ATP synthesis is linked to the development of the proton gradient across the membrane. The proton accumulates towards the inside of the thylakoid membrane in the lumen. Let's discuss the steps that help develop the proton gradient along the membrane. Water splitting takes place on the inner side of the membrane, producing protons or hydrogen ions that accumulate within the lumen of the thylakoid. The movement of electrons to photosystems enables the movement of protons across the membrane. This is because the primary electron acceptor located towards the outer side of the membrane transfers its electron to H carrier. Thus, a proton gets removed from the stroma while transporting an electron. When this molecule passes its electron to the electron carrier on the inner side of the membrane, the proton is released into the inner side of the membrane. The NADP reductase enzyme is located on the stroma side. The electrons that come from the acceptor of the electron of the PS1 and the protons are necessary for the reduction of NADP+. These protons are also removed from the stroma. Thus, protons in the stroma decrease in number, while in the lumen there is an increase of protons. This creates a proton gradient across the thylakoid membrane. The breaking of the proton gradient releases energy. The ATPase enzyme present in the transmembrane channel helps in the breakdown of the proton gradient. The ATPase enzyme consists of two parts. The F0 part embedded in the membrane, forming a transmembrane channel that facilitates the diffusion of protons across the membrane. The F1 portion of ATP synthase protrudes on the outer surface of the thylakoid membrane to the side that faces the stroma. The breakdown of the gradient provides energy to cause conformation change in the F1 particle of the ATPase. This makes the ATPase enzyme synthesize several molecules of ATP. The synthesis of ATP thus requires a membrane, a proton pump, a proton gradient, and the enzyme ATPase. Next, the NADPH and ATP produced by the movement of electrons are used in the Calvin cycle.